Hello everyone, we are now in the last part of this second lecture where we will talk about the regulation of water balance by the kidney. So let's start from an experiment, yeah? That's how I like to do things. We drink a glass of water. What happens to the osmolarity of urine? What happens to the osmolarity of the plasma? What happens to the excretion of solute in the urine? What happens to the urine flow rate? So if we drink, well, instead of a glass, let's say 1 liter, right? The plasma osmolarity, I bloody hope, stays the same in the sense that the plasma osmolarity is a finely regulated homeostatic parameter, and large changes in the plasma osmolarity are not compatible with life. I mean, you die if the plasma osmolarity decreases or increases too much. So the plasma osmolarity in a healthy individual stays the same. But following the drinking of 1 liter of water, what happens to the urine osmolarity is that it dramatically decreases within the first hour, and then within 2 to 3 hours, it goes back to the previous values, so which is the value before we drank 1 liter of water. So, what this first plot tells us is that the urine osmolarity can dramatically change so that the plasma osmolarity stays the same. It doesn't sound like a surprising thing, but the mechanism which allows this to occur is fairly complicated. So let's look at the second plot. In the second plot, we have the urine flow rate. Well, that is kind of understanding, right? For the urine osmolarity to decrease, we will have to have more water and less solute. So since the urinary solute excretion doesn't radically change with the drinking of water, what occurs is that the urine flow rate increases. And why it does increase? Because the amount of water that is being excreted increase. Okay, so following drinking water, the urine volume increases. The solute excreted remains constant in order to prevent the decrease in plasma osmolarity. Okay, so to keep the plasma osmolarity constant, when we drink water, we excrete more water. So the variation in water intake, allow our urine to change the values of its osmolarity. I mean, we are human beings, we are organisms which live on the dry land. The availability of water may not be constant, okay? So we need to be able to adapt to a certain amount the environmental conditions. So to me, this is extremely fascinating because the universe is a dark and cold place. Earth is beautiful, green, and it has the sun, but it is an hostile place. So our organism is a beautiful machine that allows us to live, to survive, and thrive in a hostile place. So, when I think about our body and I think about these mechanisms, I think about always wearing a space suit, in the sense that what the spacesuit allows us, right, is to basically keep our osmolarity in deep space, which is a very hostile environment, right? But the fact that we do not consider, I don't know, a hot beach or a cold mountain a hostile environment, or a relatively not hostile environment. I guess everything is relative in life. Some people hate the beach, some people hate the cold, but definitely it's not going to kill you, right? Or at least you can survive in it. And you can survive in it. You can survive with relative lack of water, with the relative hot, with relative cold, and so on and so forth, because of these mechanisms. So as a matter of fact, our body is a wonderful spacesuit which adapts to the environmental conditions, allowing us to survive. So this is exactly it, right? The osmolarity of urine can vary between 50 milliosmoles per liter to 1400 milliosmoles per liter, right? So if we have a lot of water, that is being drunk, then the osmolarity of urine decreases. But if we are dehydrated because, I don't know, we are in the desert, or we are, I don't know, somewhere where there's not much water, then we can concentrate our urine and save water. Now, how awesome is that? With the limit of 1400 milliosmoles per liter. And that's the reason why we can't drink seawater. And what happens when we drink seawater is that we actually lose water, because the concentration of sodium chloride in the seawater, or in general the osmolarity of seawater, is above 1400 milliosmoles per liter, which is the limit of osmolarity that urine can have. So if we drink seawater, which has a osmolarity which is superior than the maximal osmolarity that urine can have, we start to lose water. Cats don't. Cats can actually drink seawater and still gain water, because they can concentrate urine more than humans, for example. But 1400 milliosmoles per liter is still quite impressive. So water balance is regulated by three major interdependent processes. One is the osmotic gradient across the renal medulla, which can vary between 300 and 1200 milliosmol per liter. And it increases as we move towards the hills of the kidney. And that is generated by the active transport of sodium, potassium, and chloride. The second mechanism is the counter-current multiplication, and this is due to the flow of fluid in different directions in the tubule and vasculature, paired to the impossibility of water to move through tight junctions. And finally, the ADH regulation of the permeability of the collecting duct. So the more ADH is secreted by the pituitary gland, the more concentrated will be the urine. The less ADH is secreted, the more water we will lose. That's why people pee more when they drink alcohol. Okay, when people drink alcohol, at parties or at home or whatever, they urinate a lot, and the urine, if you notice, is quite clear, is almost transparent. It almost looks like water. It's very diluted. And that's why alcohol dehydrates the body, because it inhibits the secretion of antidiuretic hormone. Ethanol inhibits the secretion of antidiuretic hormone. In absence of the actions of antidiuretic hormone, we lose water, and eventually we may die, if we don't recover the water that we lose. That's why when people drink alcohol, feels thirsty at night, and that's why it's so important to drink water in case someone drinks alcohol. So step number one, the osmotic gradient. So the ascending loop of Henle doesn't have much water permeability, but it has active ion transport. So the active ion transport increases the osmolarity of the interstitial fluid, while it decreases the osmolarity of the tubular fluid. Easy peasy, okay? But then we have the countercurrent regulation. So the countercurrent regulation produces hyperosmotic gradient in the renal medulla. And that is the ability to form concentrated urine. This is due to the fact that the kidney is able to concentrate urine relative to plasma. And this is essential for survival, as I mentioned, in hostile environments, and it allows to minimize the fluid intake. The countercurrent fluid depends, on the different functions and structures of tubules along the nephron. Okay. So we have a tubular component, the descending and the ascending loop of Henle, and then we have the vascular component, which are the vasa recta and the peritubular capillaries of the renal medulla. Let's look at these in more detail. So in step number 1, and we will go through these a lot of times, okay? So in the initial phase, we have the descending limb, which has a filtrate with an osmolarity of 300 milliosmoles per liter. The interstitial fluid in white that has an osmolarity of 300 milliosmoles per liter, and the ascending limb filtrate, which has an osmolarity of 300 milliosmoles per liter. Then what occurs in the ascending limb, as we mentioned, is that electrolytes, specifically sodium, potassium, and chloride, can move from the lumen into the interstitium, but water cannot follow from the ascending limb. So what we will have as a result is increased osmolarity in the interstitium and decreased osmolarity in the content of the ascending limb. Now, to compensate that, water will move from the descending limb into the interstitium. And by moving from the filtrate in the descending limb into the interstitium, the concentration of solute in the descending limb will increase. But at the same time, new filtrate is coming down, and the new filtrate has an osmolarity of 300 milliosmoles per liter. So now the filtrate that has been concentrated is moving from the descending limb into the ascending limb. And now in the ascending limb, we have a content that has an osmolarity of 400 milliosmoles per liter. The interstitium will have an osmolarity that varies between 400 and 300 milliosmoles per liter, while the descending limb will contain both filtrates that have 300 milliosmoles per liter in the upper end and 400 milliosmoles per liter in the lower end. And now the process occurs again. So we move solutes, electrolytes from the ascending limb into the interstitium, but water doesn't follow. So we will further increase the concentration of solutes in the interstitium and decrease the concentration of the solution into the ascending limb. As a consequence, water will move from the content of the descending limb into the interstitium, concentrating the content of the descending limb. And then new filtrate at 300 milliosmoles per liter will come down, pushing the concentrated content of the descending limb into the ascending limb. And then the steps between 4 and 6 occurs until the system reaches its saturation. And the saturation level is, this is a very important value to remember, 1,200 milliosmoles per liter. This is how the countercurrent regulation works. So let's repeat it, okay? This time with writing. In step 1, the tubular fluid osmolarity is equal to the interstitial fluid osmolarity, and both in the descending and the ascending limb, and that's 300 milliosmoles per liter. Then, in the ascending limb, the active transport of sodium, potassium, and chloride allows to increase the concentration of solute in the interstitium, but since the junctions between cells in the ascending limb are tight and they do not allow the passage of water, then the concentration, the osmolarity of the content of the ascending limb decreases, while the concentration of, the osmolarity of the interstitium content increases. This leads to step number 3, where the water movement from the descending limb into the interstitium will increase the concentration, the osmolarity in the descending limb until it reaches balance with the interstitium. Now, new fluid will come into the descending limb from the proximal tubule. The concentrated content gets pushed into the ascending limb. So the hyperosmotic concentrated content gets pushed into ascending limb, and the process starts again. Further transport of sodium, potassium, and chloride from the ascending limb into the interstitium concentrates the content into the interstitium while diluting the content of the ascending limb, because water cannot flow through the tight junctions in the ascending limb from the ascending limb into the interstitium. Further water movement out of the descending limb brings to osmotic balance between the descending limb and the interstitium, and the process is repeated until it reaches saturation. So this is basically the same what I have already discussed, but it adds an element, which are the vasa recta, okay? So, of course, all the solids and the water that are contained in the interstitium will move into the vasa recta, into the vascular component of the medulla. So the blood in the vasa recta moves water, leaving the loop of Henle. And the filtrate entering the descending limb allows the movement of water, because water can move across the descending limb, while the ascending limb will move sodium, potassium, and chloride into the interstitium and into the vasa recta, concentrating, generating a progressive increase of osmolarity of the interstitial fluid and of the blood in the vasa recta, generating a hyposmotic environment of the filtrate in the ascending limb. So also blood maintains osmolarity. The medullary blood flow, to be honest, is quite low, is less than 5% of the overall renal blood flow. And this is to prevent the dissipation of the osmotic gradient that is obtained with so much work in the descending and ascending limb of the loop of Henle. The cortical nephrons contain peritubular capillaries, which remove the reabsorbed water. Okay, so that is just to remove the reabsorbed water, while the juxtamedullary nephrons contain the vasa recta, and they are the ones mostly responsible for the countercurrent regulation and the reabsorption of water and the concentration of urine. So the vasa recta carry the reabsorbed water into venous blood. So I hope that counter-current flow regulation is now clear. It's actually not difficult. It has just some moving parts. But as again, I said, it's perfectly rational. Okay, so you just need to know the different moving parts. Finally, we want to talk about ADH and its activity on the collecting duct. So I've already mentioned this, but I will talk about it again. So the tubular fluid leaving the loop of Henle, as we mentioned, has low osmolarity, okay? Around 100 millimoles per liter. So this may be lost as it is, or water may be reabsorbed more or less depending on how much ADH is produced. So if we have more ADH, more water will be reabsorbed in the collecting duct. And so the hypoosmotic tubular fluid that enters the collecting duct will be progressively concentrated, because moving towards the medulla, water will move from the tubular fluid into the medulla, following urea, concentrating the urine to the maximal value of 1200 milliosmoles per liter. But if there is no ADH, water will not be able to move from the collecting duct back into the interstitium. And so the urine will have low osmolarity, and the lowest osmolarity that urine can have is around 100 milliosmoles per liter. So how does that work? We already mentioned that, but we will talk about it in more detail. So when ADH is produced, ADH binds the V2, the vasopressin 2 receptors on the basolateral membrane of the epithelial cells in the collecting duct. These are G protein coupled receptors, specifically GS protein coupled receptors, which result in the production and the synthesis of the second messenger, CAMP. CAMP downstream regulates the insertion of aquaporins into the apical membrane of the epithelial cells of the collecting duct. And these aquaporins allow the passage of water and urea from the lumen into the interstitial fluid, which is, from the hypotonic environment to the hypertonic environment. If you don't remember what hypotonic and hypertonic means, please go back to my lectures, about homeostasis and fluid balance, and also to that nice, well, I found it nice, I had fun, workshop on molarity calculation in physiology. ADH also stimulates urea reabsorption, causing hypertonicity of the urine. So the urea movement from the lumen of the tubule into the interstitial fluid promotes even further hypertonicity of the interstitial fluid. So in that way, the antidiuretic hormone promotes the movement of water from the lumen into the interstitial fluid, reducing the loss of water by concentrating the urine. On the opposite, if we, stop the production of ADH, either because we've been, drinking too much water or we drank alcohol, what occurs is that these aquaporins are not inserted on the apical membrane, and so the water, the urea, stay into the lumen, and so we urinate, we excrete hypotonic urine, losing water. So ADH also increases urea reabsorption, okay, from the collecting duct. So urea is one of those things that we want to remove, but ADH uses urea as a trick to reabsorb more water, right? So we privilege the reabsorption of water to the excretion of urea in presence of ADH. Urea moves down its concentration gradient into the loop of L, and then it gets, once again excreted. It once again reaches, the tubular fluid. Okay, so that urea will not be reabsorbed into the blood. So even if in presence of ADH we privilege the reabsorption of water to the excretion of urea, this is only a temporary measure, because that urea is only used to increase the tonicity of the interstitial fluid in the collecting duct, but then that urea moves along its gradient and gets secreted once again. It reaches once again the tubular fluid and subsequently will be eliminated. Okay, so this is important. This is important because otherwise it seems that urea gets reabsorbed, but it's not. Okay? It actually reaches once again the tubular fluid. So this, of course, has a consequence on the plasma volume and as a consequence on the blood pressure. Okay, so the sensors in the preoptic area of the brain regulate drinking behavior. Okay, it gives us the sense of thirst. And the sensors in the preoptic area are sensitive to the osmolarity of the blood. So when the osmolarity of the blood increases, we are thirsty. Okay, so that's why after a jog, we are thirsty because the osmolarity of our blood increases. As a consequence of that, not only we have the craving for water, but we also release vasopressin and ADH. ADH, as we mentioned, act on the epithelial cells of the collecting duct to promote the reabsorption of water and decreasing, the water loss, increasing the plasma volume. Okay. On the other hand, if we have fluid intake, so we are, increasing the level of plasma volume, we want to lose that. And that is mediated by the decrease in the release of ADH due to the dilution of the reduction of the plasma osmolarity. So there is a form of diabetes called diabetes insipidus, which is caused by the inability of the kidneys to preserve water. So we frequent urinate and we are always thirsty. Okay? The diabetes insipidus means without taste, without salt, because the way, in the old times, doctors would diagnose diabetes was literally by tasting the urine. And if the urine didn't have a salty taste, then, because it wasn't concentrated enough, that may be a sign of extreme loss of water. And the diabetes insipidus is caused by a lack of ADH. And the lack of ADH can be due to a damage to the hypothalamus as a result of an accident or as a result of surgery, or it can be nephrogenic in the sense that ADH keeps being produced, but the kidneys fail to respond because they don't have the adequate receptors, or those receptors are not active to ADH. So this is different from the diabetes mellitus, which means sweet diabetes, because in diabetes mellitus, the urines are sweet because they contain glucose. The urine in the diabetes insipidus don't contain sugar. They are just very diluted. Okay? So the sympathetic nervous system, of course, plays a role. We mentioned the short, mid, and long-term regulation of blood pressure, which definitely fits with the regulation of water reabsorption. The electrolyte and water balance are regulated by sympathetic nervous system in addition to hormonal control. And it is because the sympathetic nervous system results in the constriction of afferent arterioles, reducing the glomerular filtration rate. That leads to a decreased excretion of sodium and water. And of course, the activation of the orthosympathetic noradrenergic system leads to the increase of renin release, and so angiotensin II formation, leading to increased reabsorption of sodium and water. I'm not going to get into the detail of this because you've already covered these into the sympathetic nervous system lecture. So briefly, just to show you in a diagram what I said with words, the noradrenaline released from the sympathetic nervous system leads to the constriction of the afferent arteriole, leading to reduced blood flow into the glomerular system. And that decreased blood flow leads to the increase in the secretion of renin, and so the activation of the renin-angiotensin system, which then leads to further arterial constriction. So, in this feedback loop, and also it leads to the activation of the adrenal gland, or the cortical cells of the adrenal gland, leading to the secretion of aldosterone, which promotes the reabsorption of sodium and water in the distal convoluted tubule. So, to sum up the electrolyte and water balance in the different tubular compartments of the nephron, here you have summary. Sodium is reabsorbed in the proximal tubule obligatorily, and also in the ascending loop of Henle and in the distal tubule. But in the distal tubule, it's regulated mostly by aldosterone and in the collecting duct. Potassium is reabsorbed in the proximal tubule and in the ascending loop of Henle, but it's secreted in a regulated way by the aldosterone in the distal tubule. Urea is reabsorbed in the proximal tubule and reabsorbed in the collecting duct, but be careful, the reabsorption in the collecting duct doesn't bring urea back in the blood, but it follows its gradient back into the tubular system, as we have mentioned. Bicarbonate is reabsorbed in the proximal tubule in the ascending loop of Henle and reabsorbed and secreted in the distal tubule. Water is reabsorbed in the proximal tubule in the descending loop of Henle, definitely not in the ascending loop of Henle. That's a very important point for the current regulation. And reabsorbed in the distal tubule and in the collecting duct under the control of the antidiuretic hormone. And this is it for this lecture. I hope you enjoyed it. I know I did, because I love kidneys very much. If you want any further information, a good to-go place is the Human Physiology book I've presented here, the Richards one. Thank you very much, and see you at the third lecture.